All right, everybody. Welcome to Rachel's thesis defense. So Rachel's a double major. All right. Double major in marine science, chem track, and applied mathematics. She's served in a leadership role in the geochem lab with us for 3 years now. Was in the second cohort of Scientists at Sea also. And then was also awarded the Claire Ruth Luce Fellowship as well, which partially funded the research that we all see today. So her thesis is entitled Establishing Biogenic Silica as a Proxy for Deep Sea Mining in the Clarion-Clipperton Zone in the Pacific Ocean. And this contributes to our knowledge of how deep sea mining operations, ultimately the plumes that are created on the seafloor, impact the areas around them. So definitely a timely presentation, I think. So the committee is composed of Dr. Greg Brooks, Rebecca Larson, and myself. And the format we'll start off with a presentation from Rachel, after which we'll invite questions from the audience. Once you all are done asking her questions, Pearlie and Rachel, then we can dismiss the audience and we'll proceed with questions from the committee. So without further ado, I'll let Rachel take it away.
All right, well, he already introduced me, so I don't need to read the title again. So let's just get into it. Actually start my timer too. So we kind of want to start with the big background. So why deep sea mining? The Hyman et al. Perspective breaks it into drivers, barriers, and enablers. Some of the drivers are why would we mine the deep sea? Energy demands, increasing energy demands, especially with the onset of lots of electric car manufacturing. A lot of the metals that are needed for that, we want more sources to be able to kind of increase that demand. And also land-based mining has very costly humanitarian issues and also ecological issues as well. And so those are kind of some of the reasons, like the push for the deep sea mining the deep sea is so important right now. And then barriers. So what is preventing effective environmental management? Things primarily not having enough information about these ecosystems in the deep sea. It's such a hard environment to study. There's not a lot of previous knowledge that we have, and so that is probably the primary limitation to moving forward with this, with deep sea mining, especially commercially, because at this time there's no commercial deep sea mining. And then also we don't know a lot about the potential harmful effects of deep sea mining. And so both of those really, that lack of information is that biggest barrier. And then some enablers. So what are the best practices for proper management of deep sea mining? Hyman et al. Lists 3 things. So a precautionary approach, and then an ecosystem approach. So focusing on these bigger ecosystems, how does this affect this whole region? And then the most important, adaptive management. So as we go forth with test mining, making sure that we sort of change how we do this management over time as things come up, as this process continues. So let's talk about where are we mining? So this is the Clarion-Clipperton Zone, the CCZ, in the equatorial Pacific. It's a very large region, 100,000 kilometers squared, as you can see from the top left. And then zooming in a little bit more, this is the actual Clarion-Clipperton Zone, and the individual colors are contract areas. There are currently 19 mining contracts in this region, all of which are associated with individual nations. These APEIs are in areas of particular environmental interest, so you could probably liken them to an MPA, a marine protected area, but basically they're regions set aside to study this area without having influence from mining to kind of protect that ecosystem integrity. Recently, APEI 13 was added. As you might notice, a lot of these APEIs are kind of peripheral to the region rather than in the middle of those contract areas, and so McQuaid et al. Argued that we should have one in this middle region that represents more of those polymetallic nodules and that sort of substrate. And so that was added very recently to kind of add to that biodiversity representation. And this study occurs in NORI-D, so this is our contract area. This is a very large region. It's a little bit smaller than Ireland, so for context. Okay, so then what are we mining? So this is a polymetallic nodule field. Polymetallic nodules look like this. They're basically layers of different kinds of metals. The metals of interest are primarily cobalt, nickel, copper, and manganese. You know, things used in electric cars. There's my little Cybertruck for you. Particularly topical, but those are the minerals of interest, and those are in high concentrations in these nodules, and they sit in these big fields of cobbles. This is not the Clarion-Clipperton zone, I'm pretty sure, but in the Clarion-Clipperton zone, there's like high density of them, they're very close together, and so there's a lot of these available polymetallic nodules. So then looking at kind of what are we actually concerned about? What are these mining impacts that we want to look at? We primarily take a benthic approach, so our focus is on the sea floor and sort of those impacts closer there. This is a mining vehicle. This is kind of like a schematic of what a mining operation would look like. We have three primary impacts that we're concerned about at the sea floor. So first, the removal of hard substrate. Organisms actually live on polymetallic nodules, and so if you remove them, then obviously you're losing some of that biodiversity. It is a pretty low activity region, and so organisms that grow on them for a very long time and maybe grow slowly, and so that is a really big impact. Also, from the actual collector vehicle traveling along the seafloor, you're going to have those impacts of disturbance, and also from driving and scouring with the tracks created by a vehicle of that size. They're huge, and so it has a very large potential impact there. And then finally, a secondary sediment plume from the collection of the nodules and then from driving. It's going to stir up a lot of that sedimentation, and because this is a region that is used to seeing very, very little natural sedimentation, this is our biggest concern in that environment is not used to that level of sedimentation. And so, talking about these plumes, so how do we measure the plumes? Because that is really a big part of this, and how do we measure disturbance in this region? We use what's called a multi-proxy approach. Proxies are basically ways, either chemical or physical, or even biological, they're ways to potentially use that to assess health of that region, traveling of plumes, sedimentation, things like that. We use things like sedimentology, mineralogy, so XRD, this is kind of texture of sediments. Things like phytopigments often are good for biological proxies of productivity. We use things like XRF, X-ray fluorescence, spectroscopy, for checking elements of composition, which can be useful for tracking plumes as well. And things like radioisotopes, especially thorium-234, which is a short-lived radioisotope that's very useful because it absorbs to fine particles, and so we can use it to kind of trace various disturbance in that region. And then potentially with this study, the idea is that we want to add one more proxy, and this proxy is biogenic silica. So talking a little bit more about plume composition, to orient you, this is a core from our lab. It's from a control site, STM-190. We can see that the first, it has very distinct layers, right? There's this slightly darker layer at the top, and then the lighter layer in the middle, and then this darker layer down here. So layer 2 is going to be preserved radiolarin clays, and then very deep down the core we have carbonates, but primarily we have these radiolarins. And then right at the surface is going to be this biogenic silica that we're interested in, these more recent surface radiolarins. And looking at that, let's just talk about biogenic silica in general. So how does it get there? Radiolarins, diatoms, and silicoflagellates are planktonic, and they are going to, when they die, they're going to sink, and then as they sink, they're incorporated into sediments and they're preserved there. This creates siliceous ooze, so the Clarion-Clipperton zone primarily has siliceous oozes. There are also calcareous oozes as well. Why is it siliceous and not calcareous? It's because it's below the calcium carbonate compensation depth. Basically, calcium carbonate dissolves because this equatorial water is higher in concentration of CO2. So we primarily end up with a lot of these radiolarins because that's what's preserved, they don't dissolve. And so these sediments in the equatorial Pacific are very high in them. And again, looking at the silicate water column profile, we see that low at the surface because silica is incorporated into their shells. These are living organisms, they live at the surface. As they die, the concentration increases as it becomes distributed into the water column, and then it reaches our sediments. These are some representation of radiolarians from the Clarion Clipperton zone and otherwise, and then this is a sponge as well. It's on the side of a nodule, so it's a little sponge. But those are all incorporated into biogenic silica, but primarily are radiolarians. So our driving question is, can we use sedimentary biogenic silica as a tracer for deep sea mining sediment disturbance and plume? So our goal is to add another proxy primarily. So this is a hypothesized control core based on silica dissolution and also preservation in cores previously. We see that at the surface we should see a lower concentration of silica, or weight percent of silica is our primary measurement here, because there's going to be some dissolution with that pore water that's at the sediment water interface. And then we see an increase as we get this higher signature of that preserved radiolarian ooze, very intact. And then deep below that should be carbonate sediments. We might not see this because they're very deep below, but that's the idea, is we have this pretty much preserved core, and we can understand based on that. We can check then looking at post-collector cores, so after that disturbance occurs, we think that we might see a deposition core, so things close by should have that increased signature because we're seeing disturbance of that natural layer. And then potentially in the tracks and just in general in the area, seeing scouring or removal of that surface signature based on the travel of the mining collector vehicle. So this is the kind of driving idea of, like, can we trace pre and post collector tests using biogenic silica? So let's get into the methods. Samples were collected with the RV Island Pride using a multi-core. This is the one from the RV Island Pride. There were two campaigns, Campaign 7A, which occurred in July and September of 2022. This was our baseline campaign to do before any influence, any human impact in that region. We wanted to take cores and make sure that we have that fully preserved, pristine, in theory, ecosystem. And then after, in November and December of 22, after a collector test and everything was settled, then we took cores to have that kind of comparison. How is that disturbance being in? That's sort of the driving idea behind this, is before, after. And then the samples were frozen, brought back, and freeze-dried. Pretty standard stuff. This is kind of this region looking at where these cores were taken. So in the top left, we have the control area. So these are that red box is that really far away, that 7-kilometer distance, based on this table here, how far away from the actual collector test it is. Looking over here, this is our test field. Based on our distance from the plume, this red dot is the direct, or the tract. We're going to be calling it the tract for this study, and then increasing distance from the plume. So, zone 2, 3, 4, 5. So getting away from that influence, and based on the modeled sedimentation. And so that's how cores were collected. A primary part of this study was focusing on adapting this Morlock and Freelick 1989 wet alkaline method. This is usually used in lower concentration sediments, and so we wanted to see if we can use this method and apply it to Clarion Clipperton zone sediments. Traditionally, it's a 5-hour digestion of freeze-dried sediments with a base sodium carbonate, and then we have a bonding. It basically bonds to the molybdenate complex with the silicon hydroxide and water. It kind of turns like a pale blue color, and then we measure that absorbance with a spectrometer and run calculation with a standard curve, pretty standard analytical technique. This is our standard curve. So some of the contested stuff in the field, there's 3 main things. So the length of the digestion time, the concentration and strength of the digestion solution, and then the silicate standard, so which one is used for that. And these are all contested among lots of different researchers. And so I did a literature review to kind of assess that and did various tests to see what works best. So we found that for length of digestion time, that we were comparing between a 2-hour and a 5-hour digestion. Took a lot of experiments to get it right. Basically what we found is that with a high concentration sample, a shorter amount of time it's going to dissolve in a shorter amount of time. So we get to that 2-hour digestion. Because these are such insanely high silica concentration sediments, we don't need to run it for that long. It's pretty much redundant. And with any kind of method, we want to be as efficient as possible. For lower silica samples, we find that max digestion is going to occur at the 5-hour point, so it does need that entire time. But this kind of branches the method in a way based on what sediments you have, right? And then for the digestion solution strength, this is also a little bit contested, kind of what the strength of it is and what base you should use. We use the base to dissolve the silica from the sediments, and then the debate is between strong or weak base. And so I tried various concentrations of sodium hydroxide and sodium carbonate. I found that the stronger base works faster, and it also works better for highly preserved radiolarians, which applies to our current collections of sediments. Again, this kind of goes to that branching method. Which one do you use based on your sediments? And then for silica standards, basically the issue is that there's no published silica standard. There's no highly agreed upon standard for that. I ended up just using sodium silicon hexafluoride, which is a pretty common analytical reagent, and it ended up having a fairly good standard curve. And then silicon tetrachloride is also an effective one as well, which the other standards are not quite as useful because they're impractical, expensive, and also just like unnecessary. Like you end up with similar results based on any of these. So it's better to use things that are commonly accessible. Okay, let's get into the results. So for orientation of just kind of understanding what we're talking about when we talk about these different regions, the control, collector tract, zone 2, zone 3, etc. These are actual pictures from this region after the collector test in 7B. We have our control here. You can see all of those different polymetallic nodules. That's what those little black dots are. And then for the collector tract, we see already that sedimentation, and we also see those little track lines as well we see track lines from the collector vehicle. Zone 2, again, those are polymetallic nodules, but they're covered with that sediment. Similarly, we see that impact in zone 3. And then in zone 4 and zone 5, you can see it's pretty comparable to the control. Again, you can see the polymetallic nodules, no natural sediment cover. Just to orient us to kind of what these graphs mean. So for the surface budget, we started by just running surface samples for everything to kind of see, could we kind of get this initial signature? And that is what we found. So looking at our samples here, it's grouped by distance from plume. So we have track, zone 2, zone 3, zone 4, and control, so far away. Blue is going to be 7B, and green is 7A. Already right away when we ran these samples, we could see that there's we find this higher signature, which is what we were expecting in zone 2, which makes sense. If looking at the actual pictures, we should see that sedimentation. We also see an increase in the collector tract, which again, sort of what we were expecting, at least see some kind of variation between 7A and 7B. Similar down core and further down core, signature is pretty much the same. And so after we ran these initial surface samples, we wanted to look down core because what's useful about that is we can assess what depth of sedimentation we're seeing, especially compared to the natural. Similarly grouped by distance from the plume, but these are 7A. So this was prior to any collector test. These are our baselines. We see they're pretty consistent down core, pretty low variability in the core between different intervals. We can kind of see a little bit of a maximum at 1.5 depth average, which was about what we were expecting lower at the surface than at this depth average. So they're pretty much follow the natural control, the hypothesized control cores. Getting to the more interesting stuff. So in 7B, we see already that we're getting a lot of variability at the surface. Even in some of these other regions, there is natural variability. the variability is actually from sedimentation, pretty much. So we see this increase in zone 2, this really high signature, especially relative to down core. It's a lot greater than the down core samples. And then we also see that in the collector track, which again is what we were expecting, which is good. These are 7A and 7B superimposed on each other, so we can see that all the 7A cores have very close to each other in signature. The weight percent biogenic silica is pretty consistent throughout. And then we see in 7B, the spread is really big. There's a lot of variation, which makes sense because there is that disturbance. So looking back at these, so this is a comparison between 7A and 7B. 7A is going to be the lighter color, 7B is the darker color. We see our controls kind of have some variability. This may be because, you know, they're pretty variable from each other. This is probably because you can't take a core in the exact same spot, so it's possible that there's just some of that natural variability between two spots. And so that's probably why these are not that similar. Zone 3 and Zone 5, we see that they're fairly consistent between both years. Again, you can't take a core in the exact same spot, so you're going to see some natural variability, but these are fairly consistent, which is, again, consistent with what we saw. The only thing about Zone 3 that we notice is that there is no really biogenic silica signature, but if you recall from the images that we looked at, Zone 3 actually did have some sedimentation influence, and so we'll get into that in just a second here, why that is. Zone 2, again, that increased signature, unfortunately, in 7A we were not able to collect. A core was not collected for this exact region to compare to, so it's hard to compare. But if you consider it even relative to itself and even relative to the controls from that campaign, it's notably much higher. And then for the collector track, we see that increased signature, especially significantly compared to these sites which have little variability at all. This might be because of resedimentation from natural currents or flows causing some of this zone 2 to fall into this region, so we get that higher signature. We didn't really see scouring, which may be because, again, that there's some resedimentation, redistribution of the zone 2 signature. So these are heat maps. The top is going to be 7A, bottom is 7B. We have thorium-234 on the right, and then surface biogenic silica on the left. Thorium-234, something to know about it is that it absorbs to fine clays, fine particles, which have a tendency to travel further. And so we see a larger spread of the thorium-234. You see that influence quite further from, the hotspot is actually further here as well. Whereas with biogenic silica, it's a larger particle, and so it's going to settle faster. And so we see that that's a little bit more constrained. And so that's an important distinction between these two proxies, is because they are different kinds of particles, they have different settling dynamics, and they're going to travel different distances. Now, just comparing between these two proxies now, we see a kind of higher natural concentration in 7A. I originally thought this might have been a methodological error because it's quite a bit higher than the rest of this area, even prior to human impact. But we also see that kind of increase in the thorium-234, and so it's not a methodological artifact. In fact, it is just a consistency of those natural cores in that natural area, which is good and confirms that it's a good method because we see the consistency here. And furthermore, we see the depositional hotspot. And as I said, they have different settling dynamics based on the density of the particles. And so we see that thorium goes pretty far out. Unfortunately, we did not have a zone 4, which would be right here for these cores. But even based on what we know about biogenic silica, we would know that they would settle faster. And it's likely that we would see no influence in zone 4 because we saw no influence in zone 3. In summary, methodologically, this was a very rigorous study, and so looking at a lot of these older methods and basing what we have off of previous studies that have been done, it is kind of not a one-and-done method. It basically, your method must change based on what kind of sediment you have, And so I created a branching method based on low or high composition sediments, and that can obviously be adapted further. But based on what you have to use a different base, you have to use a different digestion time. And I think having that variable method was proven very useful, and it's more efficient because we're not wasting time over digesting sediments if we don't have to. Looking more at our conclusions about the actual proxy itself and what we saw that biogenic silica is constrained spatially, and this is because it's a heavier particle, so it's going to sink faster. It's constrained a lot more than the thorium-234, which again makes sense. And then we see like a very clear influence of this sedimentation plume in the track in zone 2, but we don't see it in zone 3. Again, supporting that idea that biogenic silica settles faster. We see that also in the cores, we see that higher sedimentation is closer to the initial track, or so to the initial collector test, and we have that deeper sedimentation. It's much higher than the natural sedimentation. And finally, we see that it's consistent with other chemical proxies. I mostly talk about thorium-234, but it's also consistent with chlorophyll A increases in zone 2. But thorium-234 is a very good comparison, and especially with given heat maps, we can kind of see that graded spatial influence a lot more effectively. And we found too that the natural 7A cores are going to be consistent between both proxies. Moving forward with this project, once you create a proxy, we got to use it. And so looking forward to campaign 8A, we've started working on this already, but that is 1 year post-collector test. Now we're going to see, does this proxy, can we see any kind of influence 1 year later? How does that influence that? And just making sure that it's still consistent, still precise, still accurate. And then also applying it to other environments. So what different sediments can we use? How do we use this branching method? How does that influence what kind of samples we get? And then also comparing it with our XRD measurements of aluminosilicates. Can we back calculate, figure out what is a lithogenic silica, so something from minerals, or a biogenic silica, and kind of seeing if we can find a consistent way of calculating that and real fitting that to actual data, because this is very precise and it's pretty much the only way to measure precisely that biogenic silica, because this tends to overestimate it. And then checking it on different geoforms among other projects, because it appears to be a very precise and very accurate proxy, and so using it as much as we can. Some acknowledgements to Aiden Webster. He helped me a lot with making graphs because he's very skilled in ArcGIS. Milena Lynch for doing, again, a million dishes, and also helping with methodological troubleshooting. She was there through the rough of it, and she really helped me like bounce ideas off me and help me figure out what was wrong with our method here. And then Dr. Schwing for his supervision, and Brian O'Malley for being a reader and editor, providing pictures, general mining knowledge. He's the expert, so he knows lots about mining, and that was very useful. As Professor Schwing mentioned, this research was funded by a grant from the Claire Booth Luce program. I would not have been able to do this thesis research without that funding, and I'm very grateful for that. And then these are our affiliates, NORI, TMC, and obviously Eckerd College. And that is all I have for you. Any questions?
Questions from the audience?
So for locations that are above the CCD and do have the calcareous ooze, is there enough trace like silica in that ooze so that you can apply this proxy to those locations, or are there other methods that are used for places that do have the calcareous ooze?
That actually is a great question. So there is going to be, so in the equatorial Pacific, we have primary siliceous oozes, right? But in obviously other regions, that's going to depend based on, the water types and the water masses. So you're going to have different concentrations. This method has been used in lots of different places. And so you should be able to use it because you'll get those really trace amounts of silica, right? So you should be able to directly measure it. There's lots of ways of measuring calcium carbonate. I think primarily XRD, but I'm not 100% sure on that, but yeah, that's a great question. So you can use it in a lot of different environments.
On your graphs of the depth profiles, there were error bars. What was the measure for your error?
So I ran three replicates, like 3 analytical replicates. So 3 methodological replicates, which are different. So I basically digested the same sediment 3 times. And
Well, that was actually my question,
Yeah.
if I may interject. So when you say 3, so the carousel comes off the ship, and it drops, and it
Mhm.
looks like it has many tubes on it. Is that right?
yeah.
And so is each tube a replicate core? And so when you said you did 3 replicates,
Yeah.
are they from 3 separate cores or 3 subsamples from a strata in a core?
Yeah, so we take a sediment core, we section it out, right? So maybe we'll start with like a surface interval or whatever. So like a 0 to 0.5 centimeter interval. That's freeze dried and that sample is sectioned. So to run then the biogenic silica procedure, I basically took 3 separate falcon tubes, put it the same amount of sediment in each one, and then ran the method
From the one core?
from the one core and the one interval.
Yeah.
So they're replicates from that actual.
Pseudo-replicates.
Yeah,
Well, okay, you said that.
yeah,
Let's do
Okay,
it.
well, that's why,
yeah. So it's not like there's
I
multiple
sample in
cores.
soil and I know there's a lot of heterogeneity and your error bars are so small, they're spectacular. And
Yeah.
So is that standard error, standard deviation?
It's standard deviation.
That standard deviation
Yes.
is tiny?
Mm-hmm. Yeah, I got relative error of less than 1% for almost all of the samples.
I mean, since they're really subsamples from one core,
Yeah.
I can see now why they're smaller.
Yeah, because a lot of them are. The primary thing with this method was actually because as an analytical chemist, right, we're looking to see if the method itself is precise, right? And a lot of this study was really focusing not so much on, yes, it's important to do replicates of samples, but when you're doing an analytical method, it is important to real fit that and try to make sure that you have as close to that precision as possible. And so that was a large focus
So really,
on
like
running
if you're
multiple-
publishing, you call them subsamples rather
Yeah.
than replicates.
Yeah.
Do you have replicate cores so that you could see heterogeneity, or you just got that one core? Did they give you other ones?
For certain sites, we have replicates
Yeah.
for different.
I mean, I'm sure it'll take a
Usually
long
for
time
different
to go back
analyses.
and
So.
do the same
Yeah.
on other
Yeah.
cores.
Yeah,
but
'cause sometimes-
variability.
Because like a multi-core collects obviously a lot of cores all at the same time, right?
Right.
But we take individual cores for different analyses. Like we might freeze dry one for thorium, take some for, I don't know, foraminifera or something, freeze some for phytopigments. So they all have to be treated differently, and so you end up kind of losing that at least replicate because you're using it for different chemical analyses. And we have such a limited time out there to get those cores, it kind of limits and also it's a very deep area as well, so.
Other questions? Okay, thanks Rachel. Okay. So at this time, we'll go ahead and dismiss the